introduce Dr. Linxing Yao, who serves as an expert in targeted small molecule analysis using liquid chromatography, tandem mass spec, and GCMS. And with that, I'll turn it over to Linxing. Thank you, Linxing.
Thank you, Corey, for the introduction. So good afternoon. I hope everyone enjoyed a great holiday break. I'll start today's talk, targeted small molecule analysis, with first with what is small molecule analysis. This is a type of analysis that measure low molecular weight metabolites that are present within a cell, tissue, organism, biofluid, and other biological samples. This small molecule and their interactions are known as a metabolome. Metabolome is the result of complex interaction between genome, transcriptome, proteome, and the environment. The potential size of the metabolome can range from 600 metabolites, such as in yeast, to 200,000 metabolites in plants. The concentration of metabolome also has a wide range that can be 5 to 6 orders of magnitude. Example of small polar metabolites include small polar metabolites like organic acids, amino acids, simple sugar that are involved in glycolysis, TCA, and other pathways in the central carbon metabolism, as well as lipids, hormones, neurotransmitters, drugs and their metabolites, gut microbial products. So the research strategies for metabolomics have been divided into two approaches, targeted and untargeted analysis. Untargeted metabolomics is the comprehensive analysis of all the measurable analytes in a sample, including chemical unknowns. In targeted analysis, there are only a limited number of metabolites analyzed. So the focus of sample prep should be on the needed metabolites while largely remove interferences. So the extraction method is usually customized. Targeted analysis can do both absolute and relative quantification. In order to obtain absolute quantification, you would need to use internal standard and calibration curve. And there is also a growing trend to use internal standard in untargeted analysis to improve the data quality. So targeted analysis does not need annotation compared to untargeted analysis because we already know what we are to measure. It's more suitable for hypothesis-driving studies where you already had a list of compounds that you want to know their changes in response to treatments. It's also the preferred approach for analysis needing high sensitivity because of the instrument it uses. So the commonly used instruments for running target analysis include LC, tandem mass, and GCMS. At ARC, we have Waters Acuity UPLC coupled to Xevo triple quadruple, and also Thermos GC coupled to single quad. For today's talk, I will only focus on the LCMS and how it is used in our targeted analysis. The LC tandem mass system start with LC separation, followed by ionization, and then being analyzed by mass analyzer, and then detection. The last three steps all happen on mass spectrometer. So then, next, I'll briefly go over each in the next few slides. LC stands for liquid chromatography. Its separation is resulted from the partitioning of analytes between the mobile phase and the stationary phase of a LC column. Commonly used column chromatography include reverse phase chromatography and hydrophilic interaction chromatography, as we call HILIC. Reverse phase column, polar analytes elute first, and non-polar analytes will have stronger retention on the column. HILIC is on the opposite, so these two column chromatography are complementary to each other. The output of LC separation is reflected as analyzed peak appear at different retention times, which is determined by their molecular structure, dimension of the column, type of the chromatography, as well as mobile phase and gradient program. After the sample goes through the separation through LC column, it's then sent to the mass spec. In order to be analyzed by mass spec, the molecules need to be first ionized to charge the ions. Electrospray ionization is one of the most important ionization techniques and most commonly used for LC-MS. It's suited for a wide range of applications, especially for the polar molecules. So ESI is called as soft ionization techniques as opposed to the hard ionization techniques used on GCMS because there's little fragmentation happen to the molecular ions during ESI process. The first step of ESI is the dispersion of a liquid into electrically charged droplets. Samples from the LC are introduced at atmospheric pressure into the ionization source. A high voltage is applied to generate a fine spray of charged droplets, usually with the assistance of nebulizing gas. The charged droplets pass down a pressure gradient and a potential gradient towards the mass analyzer. During this travel, droplet size decreases and charge density at the surface increases as a result of droplet disintegration and solvent evaporation. Finally, when the electric field at the surface has become sufficiently high, ions are emitted into the gaseous phase and are sampled by the mass analyzer, which is under high vacuum. This slide shows the ion optics inside the mass spec. So after ionization, the charged ion pass through the sample cone, which is here, or you can see from this picture, into the ion guide. Which is here and here. In Waters instrument, the ion guide is called step wave. So the ion guide focuses the ion beam, remove the neutral species, and then direct the ions to the first quadrupole, which is here. That's shown here. And then the second quadrupole is actually not a real quadrupole, it's a collision cell, where the ions can undergo collision-induced dissociation or in a simple words, fragmentation. And then the ion passes through the third quadrupole, so over here. And both first and third quadrupoles are used to filter the ions according to their mass to charge ratio, m/z. At the end, the transmitted ions are detected by a photomultiplier detection system. We'll talk more about this quadrupole region next. So first, let's go over some basic terminology. I'm using lysine as an example here. So lysine is an amino acid with a molecular weight of 146. And if it's ionized in positive mode, it will become a possibly charged ion with m/z 147. So this is called a precursor ion or parent ion. If this ion loses a carboxy group or amine group, it will become a fragment with m/z 101 or 130. If it loses both of this polar group, it will turn into a fragment with m/z 84. So these fragments here are all called product ion or fragments ion or sometimes daughter ions. So you may have seen the term MRM, SRM in publications. So MRM stands for multiple reaction monitoring, also known as selective reaction monitoring. It's very commonly used method for quantitative analysis when you use tandem mass spec. First, a complex mixture of ions enter the first quadrupole and filtered based on their m/z corresponding to the precursor ion of compounds of your interest. In the case of lysine, it will be 147. And then this specific precursor ion 147 is fragmented in the collision cell. And the generated fragments are transferred to the third quadrupole, which selectively detect product ion of your interest. And in this case, lysine, it will be 84 and 130. So you see, only compounds that meet both criteria, specific precursor ion and the specific product ion, are isolated within the mass spectrometer. By ignoring all other ions flows into the mass spec, the experiment gains sensitivity and selectivity. So the pair of precursor ion and product ion are called as MRM transitions. So lysine has 2 transitions here. So how do we set up MRM method on the instrument software? So I have this interface. This interface is from Waters Instruments MassLink software. So each line item here is a function of our MRM method. Within each function, you can build one or more MRM transition. It could be for one compound. One compound has 2 transitions, or it could be multiple compounds. And for each transition, you want to specify it's a precursor ion, product ion, as well as Coulomb energy and collision energy. We'll talk about this later. And you want to specify this MRM method, is it for positive or negative ionization? And also you want to specify the time window when the MRM within this function will be scanned. And then if you go back to this main page, each green box, the size of the green box represents the time window for each function. And then lastly, we also need to specify the dwell time. Dwell time is the amount of time that the instrument measures one transition. It correlates to the number of data points measured over a chromatography peak. It's very important for quantitative analysis to obtain a proper number of data points across a peak, because the data point define the peak shape, and therefore determine the accuracy and precision of peak area calculation. The rule of thumb has been 12 to 20 points across a peak. When I prepared the slides, I came across a recently published paper. The authors found that when you have a really narrow peak, usually this can be achieved on UPLC, like the instrument we have. You can use as few as 7 points to get the same accuracy and precision as when you are using more than 20 points across a peak. So after data acquired, you can look at the data using the instrument software, or you can use Skyline. This is widely used as open source software. You can check on the Skyline software website. They have a nice tutorial to get you through to learn how to use the software. It's easy to use, and you can check the peak shape and retention time matching. Here, each peak, the different color of the peak represent different MRM transition, and you can look at the peak area calculation for each peak. And if you want, you could build the program in Skyline to do all the calculations with including calibration curve fitting and final concentration calculations, and then export this to Excel. So we talk about using at the beginning of the talk, we talk about using internal standard in target analysis in order to calculate absolute quantification. The other reason that we want to use internal standard is to use it to normalize for analytical variation resulted from sample prep and instrument signal drift. So how do we choose internal standard? Well, first, we need to look at the stability and performance. In general, the stable isotope labeled standards are preferred over structure analog, because they behave in the same way during sample prep and data acquisition. And usually carbon-13 is better than deuterated, it's because it's more stable. In return, it's also pricier than the deuterated, and less the availability of C13 standard is not as good as the deuterated standards. The ideal concentration of internal standard should be approximate to the average analyte concentration in the sample set, or sometimes you can use lower concentration if your label standards are really expensive. And when testing internal standard, you also want to watch out for interferences from impurities from the internal standard and those naturally present isotope. So the example I'm giving here is plant cannabinoids assay we developed at ARC. In this assay, in this panel, we have two compounds, CBG and CBD. We cannot separate them by LC chromatography. And these two compounds their masses are only 2 units difference, 315 and 317. But if you look at their other isotope, their M+1 naturally present isotope are present at non-negligible abundance. So which means that if I want to use a CBD-D3, this is the very few available standards for this method. If I use CBD-D3 as internal standard, which we're going to measure 318, we'll get a signal, CBG 318 will contribute to the signal of CBD-D3. And what even worse is CBD-D3, the deuterated form usually eluted slightly earlier than the non-labeled analytes. So that means its peak is going to be even closer to CBG. And you will see, when you measure CBD-D3, you will see a peak or maybe with a bump or on the shoulder of the peak that you cannot distinguish between the signal of CBG and the CBD-D3. So the solution is, instead using CBD-D3, we switch to THC-D3, which has the same precursor ion 318, but its retention time is away from this group of compounds. So how do we use internal standard to run calibration curve and calculate absolute concentration? Assume that we have a sample set that we want to measure lysine in this sample, and we know that the expected concentration is about 0.5 to 3 microgram per ml in this data set, in this sample set. So then we can prepare a serial diluted curve that you may use a 4 microgram per ml as the highest concentration, and then dilute it down to 0.25, which covers the range of the expected concentration. And then you add internal standard, same amount of internal standard at the very beginning of sample prep, before sample prep, to sample and to the curve sample. And after that, you treat the sample and curve in the same way during sample prep and during data acquisition. And then in the data acquired, we can get a peak area of lysine and peak area of internal standard in the curve injection, as well as in the sample injections. So you take the ratio of peak area of lysine to the internal standard, this will be the Y, and you plot it against the X, which is the concentration of lysine in the curve sample that you already know when you prepare it, and to fit to a linear equation. And down to the sample set, to the data of in the data from the sample injection, you can get this same ratio, normalize the peak area from the data, and then you take this back to the equation to calculate the X, which is the unknown concentration of lysine in your biological sample. So the calibration curve is a relationship between the instrument response and the concentration of analytes. If there is more than one analyte, a calibration curve should be generated for each analyte. Do we always want to use internal standard and the curve for targeted analysis? So it depends on the scope of the project. If you are only interested in group comparison, fold change, then you don't need to use a calibration curve. But it's good to have internal standard there to normalize for the analytical variation. But if you need to know the absolute concentration, then yes, you need to run the calibration curve as well as spiking the internal standard. The cost of spiking internal standard is only related to the price of the standard itself. But running curve means extra instrument time, extra prep time, and data processing time. So it may become expensive for running the curve if you only have a small project with 4 or 5 samples in there. So the method setup for targeted analysis usually requires method development to different extents, more time spent upfront. Sample prep and instrument parameters need to be optimized to achieve desired separation and sensitivity. Tweaking on internal standard and curve fitting, as we just talked about, is needed. And then we also want to test the carryover. So, and there are certain criteria and metrics that need to be met depending on the research need and as well as the type of analysis. So for specificity, it is the selectivity of the approach, identifying sample with isotopic, isobaric interference, and thereby providing confidence in concentration assignment. We could use retention time, peak shape, MRM transition, as well as the transition ratio to evaluate this method specificity. So here's an example that for oxylipin panel that we developed in collaboration with Dr. Tara Norgram. So oxylipin is a group of bioactive lipids which help regulate inflammation and oxidative stress found in all tissues, urine, and blood. It is an oxidation product of polyunsaturated fatty acids. An example is here. So we have fatty acid with 20 carbon chain lengths, and it originally has 4 double bonds, except one of the double bond is oxidized to epoxy group. So in this assay, we have 51 target and 13 internal standard. The challenge is the separation of isomers. The list here represent a subset of the panel, and the color blocks indicate the isomers. So we have many positional isomers in this panel. For example, this group at the bottom, that's a 6 compound that have the same molecular weight, same carbon chain length, and they all have one hydroxy group. But it's at a different position on the carbon chain length. So we need it for this kind of method. This kind of analysis, we need to pay special attention to the method specificity. First of all, we need to enable the best possible separation on the LC column. So we tried a column of different dimension, try different buffers and gradients, which resulted in the chromatogram presented here. Now, next slide, we're going to zoom in to look at this 6 compound, how they look like on the LC, on the chromatogram. So these are this 6 compound. 2 of them can be separated from the rest of them, but the compound with a peak number 14 to 17, especially 14, 15, 17, they are overlapped. 16 is almost separated from the rest of the group. This is how the best we can get from the LC separation. The next step, we can look at whether we can find a unique MRM transition to further separate those overlapped peaks. So for peak 14, for example, we found a unique fragments 155 that's not shared by any other peak. So that means we can use this MRM channel to specifically quantify peak 14, even though it's almost a complete overlap with number 17. And then for peak 15 16, they share the same fragments 167, but because we already separated them on LC on the LC column, so that means we can use the same MRM transition to quantify this two peak, and then as reflected on the result, you will see the two distinct peak, not interfere with each other. And we have, so there's only number 17 left. Unfortunately, number 17 shared the same fragments with number 15, that is 179, and it's not, it's 50%, it's only 60% or 40% separated on the LC column. So when we run a real sample that has both number 17 and number 15 present in that sample, you will see a peak. You're probably going to see a double peak like this, and then the part of this is from 17, but the other part of the signal is contributed from compound 15. But there's other way we can work around this. For example, we can use a peak height to calculate instead of the peak area. Okay, so for precision and accuracy, precision is the indication of the repeatability of series of measurement. We usually use a coefficient of variation to evaluate it, and accuracy is the deviation from of the measured value from the true value. These two metrics can actually evaluate it using one same experiment, where you can prepare a same sample but spike different amount of target analyte standards into the sample. So then you have low, mid, high concentration of targets in different samples. Let's say three levels concentration, and then you analyze this three samples in replicates and at different days. So then you can get CV of interday CV and intraday CV. The recommendation is your CV generally for technical analysis should be below 20%. And for accuracy, since you already know how much spec in each sample, and you can compare it with the theoretical value to the measured value from all these replicates. For analyte stability, we want to look at how the raw materials storage affect its stability. And then how freeze thaw affects its stability before extraction. And after extraction, will the sample extract be stable while it's sitting on the autosampler? And will the sample extract stable over the time when stored in the freezer and can be reanalyzed if needed? And if diuretization is involved during sample prep, we also want to evaluate the diuretized sample, its stability. So I have an example here, is a method developed for measuring NAD and it's metabolized in whole blood. So NAD is an essential cofactor in cellular redox reaction, plays critical roles in energy metabolism, and NAD and its metabolites including several nucleosides and nucleotides are referred to as NAD metabolome. They are critical mediator in the aging process. The challenge with this group of analytes is that they are highly labile. So first of all, we have several redox pair in the group, and also the larger molecule in this group are prone to degradation, and their degradation products are also the target in this group, in this method. So we have to look at their stability on the autosampler. So as expected, the reduced form of those larger molecules like NADH, NADPH, as well as their oxidized form they degraded rapidly when sitting on the autosampler. So that means we have to control the time, how long this extract need to be analyzed after they are after the extracts are ready. How soon they need to be analyzed to keep the data more consistent. And then we also look at the freeze-thaw, how the freeze-thaw affect the stability of some of the compound in this group. So our standard procedure is adding, we started with frozen whole blood. Our standard procedure is adding organic solvent to the frozen sample. And during thawing the sample, immediately in contact with the organic solvent, the enzyme get deactivated. That's the way we can minimize the enzyme induced degradation. Then we compare our standard method to the sample that thawed before adding organic solvent, and compare to the sample that accidentally suffer 1 cycle or 2 cycle freeze-thaw before the extraction. And you can see how much the freeze-thaw and thawing detrimentally impact the compound stability for the larger compound. And in response, you see an elevated concentration of the degradation product NAM in those samples that's subjected to freeze-thaw treatments. Okay, so linear dynamic range can be determined by running calibration curve over a wide range of concentration. We need to know at which point the signal response is no longer linearly correlated to the concentration. Then we can modify our method by using a smaller amount of starting material or do dilutions. Triple quad instrument generally can maintain a linear range of 3 orders of magnitude. The limit of detection, LOD, is the minimum sample concentration at which the analyzed signal can be reliably detected when compared to the baseline noise of a Planck run. A signal-to-noise ratio 3:1 is generally considered acceptable for estimating the LOD, and 10:1 for LOQ. These two, the LOD and the LOQ, can be calculated and defined in different ways. For example, the Clinical and Laboratory Standards Institute defines LOQ as the lowest amount of analyte in a sample that can be quantitatively determined with a stated precision. So it could be, the stated precision could be 20%, 30%, or 35%. The lack of clarity in defining LOD and LOQ can be problematic, especially in interlaboratory study or in the study where presence-absence determination is crucial. So it is important to clearly define the LOD and LOQ for each method and be consistent. Sensitivity. So one of the triple quad instrument, one of the advantage of triple quad instrument is the high sensitivity it provides. However, it takes some effort to achieve the best sensitivity of a specific method. There are a few approaches that can be used. The example given here is a method for measuring blood CBD, THC, and their metabolites. We have seen a growing number of labs that are interested in the quantification of these compounds in blood for studying their metabolism and the pharmacokinetics. 7-carboxy CBD is the most abundant CBD metabolites in the blood, and 11-hydroxy THC is psychoactive, is at least equal potent to THC, but more readily diffuse into the brain than THC. So this figure shows the comparison of each compound with and without SPE cleanup during sample prep. So you can see there's a huge matrix effect that caused ion suppression when not using SPE cleanup, which is the yellow series. With the interference removed or partially removed, the sensitivity was improved a lot. And it's also interesting to see that the signal increase after SP cleanup varied with compounds. For like the analyzed with carboxy group were less impacted by the SP cleanup compared to others. We also earlier when we talk about MRM method setup, we said that we need to specify the cone and the collision energy within the MRM method. So cone energy can maximize precursor ion transmission from the source to the next stage of ion optics and can minimize in-source fragmentation. Collision energy can help optimization can maximize the generation of product ion. So if you test, for example, if you test the collision energy in a systematic approach by increasing the voltage, you will find the highest signal intensity can be obtained at certain voltage, which then is the optimum collision energy for this MRM transition. Same observation for cone, but the cone energy is not as a clear trend as the collision energy. We can also improve sensitivity by making the ionization higher by enabling higher ionization efficiency. So in this example, this is a recently a method we recently worked on. So this is a group of compound that belong to steroid hormones. The figure on the left shows how the steroid signal change with the type and concentration of LC buffer additive So the light green is using formic acid as additive. The darker green is using ammonium fluoride, and the yellow one is also ammonium fluoride, but at 10 times higher concentration. And okay, so you can see how the signal changes with the additives. The exception in this group, or the challenge in this group panel of compound, is estradiol, which is a female sex hormone, and its physiological level is extremely low in certain population, such as in male and in postmenopausal women and in children. So we need a much higher sensitivity as compared to other steroids in the panel. And then we see, but however, this compound estradiol, it can hardly ionize because it doesn't have a polar group that can be easily ionized. And if we test it in formic acid buffer, the light green in this figure, we cannot get any ionization. If we switch to an alkaline buffer using ammonium hydroxide as an additive, we get a little bit ionization. And using ammonium fluoride, which is the best buffer in this testing, we got some promising result, but still the sensitivity is not good enough. So we turn to diuretization approach. We add a diuretization group that can be easily that is more easily ionized in the instrument. So then, when we run this diuretized sample in the formic acid buffer, we got a substantial improvement in the sensitivity for these compounds. Matrix effect again. So earlier, in the earlier slide, when I talk about the blood CBD THC signal experiment, and where we see a negatively impact from sample matrix. That's the ion suppression from the sample matrix. Here I have an example because the matrix effect can be can cause ion suppression or ion enhancement. The example here is ion enhancement we see in when we are analyzing short chain fatty acid in human plasma. So the circle theory here in both chart represent the analytes in plasma sample, and the triangle series in this figure are the standards prepared in pure water. And you can see we got a higher response in the sample matrix than in the neat solvent. So the sample matrix effect could be from sample matrix, and it also can be from contaminants like detergents, or it can be from some non-volatile salts that added during the experiments. To evaluate the matrix effect, you want to compare the standard in neat solvent with the standards with the sample that having standards added to the matrix post extraction. So at the end of this talk, I would like to present a few examples of established targeted assay that are available at ARC. So by full service, it means that we take care of everything. We do from sample prep to data processing, and we return a Excel table of analyte concentration back to you. And we have worked with various sample matrices, broadly speaking. In this, method development is also available as a service. And if we are the typical turnaround time for a reasonable sample size like 100 or smaller sample size is 4 to 6 business weeks if this is an established assay. So I encourage you to check out our website to learn more about the targeted small molecule analysis that we can offer, and don't hesitate to contact us if you have the research need in this area. I think we're okay. So I would like to thank you for your time, and seems like I have plenty of time left for questions if you have.
Thank you, Linxing. That was great. We'll open it up for questions. I see a hand raised from Paul.
Hi, Linxing. Thank you for the talk. It was very informative. Can you hear me? Can everyone hear me?
Yes.
Yes, I can hear you. Thank you for the comment.
I was curious about the example about different steroid hormones and estradiol, how it seemed like optimizing both signals actually utilized sort of different chemistries. Like the formic acid was the best additive for the estradiol, but the ammonium fluoride and whatever. So like, how do you deal with those kind of problems when you have, you know, different conditions in the same run that are more optimal? Oh,
that's a good question. So just for this case, I can't speak in general because it's a method all different. We have to, so it seems like ammonium fluoride is working best for the 3 steroids that doesn't need the extremely high sensitivity. And it also not hurting that additive is not hurting the estradiol after diuretization. So we can choose this buffer for all the analyze. And then we also, because your question is more like you want to run everything in one run instead of separate them. So we also test if we take the whole sample, the extracted plasma sample, undergo diuretization, will that specific diuretization work for estradiol, damage any of the other steroids that's not going to be diuretized? We check that, and then we has confidence that the other compound won't affect it. So we can combine this whole steroid panel into one sample prep, one analysis using both ammonium fluoride as a buffer and diuretization in the middle of the sample prep.
Cool.
It needs some testing
Yeah.
to explore the best options. And the goal is always to save time
Sure.
and cost.
Well, thank you.
See another hand raised from Frank.
I think you're
Frank, you're muted if
muted.
you're.
Yeah. Sorry, I was trying to multitask here. Yes. Can you tell me more about why some additives really increase the sensitivity to some of the detection of some of the metabolites?
Well, I don't know exactly the theory. This is for example, ammonium fluoride is not commonly used in the lab because this is a toxic compound and it's corrosive. But there's an increasing trend that people are it's finding more and more cases that the compounds actually improved, the ionization improved by this additive, but it doesn't work for all the compounds. It's case by case. And for example, for estradiol, it's actually just a little bit improvement compared to the ammonium hydroxide additive we use. And we know for a compound like estradiol that has 2 hydroxy group, one of them is a phenolic group, that they don't behave well in the ESI ionization, but you usually can get better result with negative mode ionization. It's
okay, and then when you use those, are you looking at still M+1 or M-1, or you're looking for M+ ammonia as a MRM?
If it's ammonium hydroxide in the negative mode, I'm looking for M-H. And if it's a formic acid as additive, it's a positive mode, I'm looking for M+H. If this is also depend on the instrument. Some instruments give you stronger response of the IDAX, like a plus sodium plus potassium. And for this, and then for example, if I run the same standards on the TOF instrument in our lab, I see a lot of sodium products of it. But when I run on this tandem quad instrument, I don't see much additive. It depends on the instrument, I'll say.
Okay, thank you.
Thank you.
I see a hand from Jacob.
Excellent talk. Have a quick couple of questions for you. The first one is, are quality control samples often used for LCMS analysis, and what role could that play potentially for LCMS? And then out of curiosity, what matrices have you found to require the most method development for analyzing them?
First of all, the quality control, yes, it's implemented in all the analysis at ARC full service projects. So we generally use a pooled QC, which you take a small amount of sample from all your study samples, and then so that's the best represented what compounds in you can see there, and then the concentration. And this pooled sample we injected after a few injections, let's say every 6 injection we inject the QC, and then during data processing we evaluate the CV of the QC to indicate, it's an indication of how the instrument stability was during running the sample sets. And also, like I mentioned, the internal standard is also a good solution for quality control. You can see how the signal intensity of internal standard change over time, and you want to expect a consistent value from internal standard. And as far as what is the most difficult matrices, that's all the matrices are difficult for targeted analysis. Especially when you are analyzing a compound that you need an extremely high sensitivity, then the matrix effect become significant. But if for example, we are analyzing amino acid in a sample, it may not be a huge concern because the concentration of the analyzer is high enough for us to ignore the matrix effect, and we have internal standard to correct for any suppression if you have a good internal standard to correct for the difference in ion caused by the ion suppression on different compounds.
Awesome, thank you.
Okay.
I see a Joe with a hand up.
Hey, yeah. Hi, Linxing, and thank you for this kind of excellent presentation. I just got one questions on the potential impact of type of instrument on the sensitivity of your different type. Generally speaking, the different type of analysis is quantitative analysis. So I can see we may use a triple quadrupole or QTOF and for targeted analysis. Is that
Yes,
correct?
in theory, you can use both. And if you're craving for the highest sensitivity, triple quad usually is the better than TOF instrument, at least at the current level of instrumentation configuration. And the other advantage with triple quads is you can, and so both TOF and the triple quad can run MRM experiments. But with triple quad, you can include a lot of like 100 compounds within one method. And then this is not doable in TOF. Because it is related to the scan time, for a certain time, how much you can do for how much you can scan.
I see.
How many
So
compounds you can scan.
Yeah, generally speaking, that means as to the number of the analytes you're going to do, or the sensitivity. I mean, the triple quadrupole could be a better choice, right? In this case.
Yes, most of the time, triple quadrupole. If you have that instrument, that will be your best option. And if you're only interested in one compound and you don't worry about its sensitivity, its concentration, is it good enough to be on TOF, then either one, whichever is available, you can use.
Generally speaking, do you think could you have some kind of really a rough estimate or how much or the sensitivity could be higher and from the triple quadrupole than the TOF? Or do
I don't.
any...
I see Corey nodding his head. I don't have a good number that I can be backed up, but I feel like at least 10 to 100 times. That's my feeling.
I see.
My benchmark is for a comparable generation of instrument, comparable model, 10-fold is 10 to 20-fold is probably a good benchmark, good rule of thumb.
I see. All right. Yeah. Thank you both.
Thank you.
We do have a couple more minutes for questions. If anybody else has a question, feel free to chime in. Okay. Well, I think we will stop then. Please join us again next time. Dr. Gustavo Diaz talking about metaproteomics, and then Dr. Claudia Boot talking about self-service mass spec in ARC. Thank you all for attending. Have a great day.
Thank you. Bye.